Hello, everyone, and thank you for being here. This is the second of three installments in the Beyond Mammals Exploring Cancer Models series, with this webinar being focused on the model organism Drosophila, the fruit fly. I'm Justin Benavides, a program director at NCI's Division of Cancer Biology. A link to live captions can be found in the chat if you need those. We have a full agenda today, but first, some quick housekeeping. We will open this webinar with 3 talks from our talented speakers. Please hold your verbal questions during this part of the webinar. If you'd like, you may enter questions in the chat box and we will get to those following the talks. Once the talks are finished, we're going to start with the discussion session. I'll open up the discussion with a couple of standard questions, and then I'll open the floor to the rest of the audience. During that discussion, feel free to either put your questions in chat or raise your hand so that you can unmute yourself for a verbal question. Please take care and help us to create an atmosphere of mutual respect and sensitivity during the discussion session. Finally, I'll say a few words about the series, and I'll say this at all of the installments of the series. Here at the Division of Cancer Biology, we are interested in the landscape of cancer models. Much of the research funded by our division is in mammalian models that we all know and love, like the mouse, and a growing proportion is dedicated to developing computational models. But the modeling landscape is wider than just those 2 examples. And in this series, we hope to highlight the work of researchers who use non-mammalian animal models like the fly to answer cancer biology questions and to start a conversation about cancer modeling more broadly. So without further ado, I would like to introduce Dr. David Bilder, professor at the University of California, Berkeley, Dr. Norbert Perrimon, professor at Harvard Medical School, and Dr. Wumin Deng, professor at Tulane University. Take it away, Dr. Bilder.
All right. I am going to share my slides. There we go. Can everybody see that?
Yep, looks great.
Okay, let me get a laser pointer. All right. So thanks everyone for coming to attend the seminar, and thanks Justin for organizing this very important webinar series. What I want to do with my time is not to tell a single scientific story from my lab, but I think the biggest impact is going to come if I really start out by making the general case for the fly as a complementary animal model to study cancer biology that has some really unique advantages. And I want to do this in the beginning by using a slide that I use when I teach undergraduates. And this slide is really meant to emphasize the fact that some of the most important discoveries in cancer biology have come from very unexpected sources. And so this slide is about the discovery of oncogenes. And the discovery of oncogenes begins when a Long Island farmer brings a hen, a chicken, to Peyton Rouse at the Rockefeller Institute. And Rouse realizes that this chicken has a growth that's actually a tumor in a bird. And he isolates the transforming principle from this tumor. And that then allows Howard Temin and Harry Rubin to develop this focus assay, which is essentially like making cancer in a dish. And what I really try to convey to the undergrads is the incredible imaginative leap that you need to make between, a bright spot in a petri dish and the tragic, clinical progress of cancer that you see in a patient or maybe even in a loved one. But it's exactly that imaginative leap that is critical for making molecular mechanistic progress, leading to the cloning of the oncogenes, the discovery of their enzymatic activities, and therefore the design of rational therapies. Okay, so at this point, we really still need animal models. We do not know enough about a cell, much less a cancer cell, to be able to use kind of a virtual cell or anything like that to solve cancer. And so for many decades, the most prominent cancer model has been the mouse. And the mouse is a great model for cancer because it's a mammal that's closely related to us. It has many advantages and has made very important advances to our understanding of cancer biology. But it's important to understand that it's not a perfect model. It has some disadvantages as well, and I've just listed those here. And so in my lab, we've been very interested in the idea that since mice are not a perfect model for human cancer, are there other animal models that are perhaps more distantly related to humans that might also be useful models and might have unique experimental advantages that could lead to really different perspectives and different kinds of discoveries. And so the animal that we have chosen, of course, for the seminar today is the fruit fly Drosophila. Drosophila is an insect, and therefore it's an invertebrate. So it's more distantly related to humans, but it has this incredible track history over 100 years of absolutely fundamental foundational advances in biology, 6 or 7 Nobel Prizes, and that these advantages, which include its rapid generation time, its very low cost of care, the fact that we can generate large numbers of these for statistical confidence and significance, the fact that the ethical regulations on invertebrate animals are less stringent than those on animals like mice. And finally, just what's often called the awesome power of fly genetics, the ability to manipulate very rigorously and precisely gene expression in multiple tissues at the same time. That all of these experimental advantages together could give this complementary perspective that could lead to the kind of transformational advances that we're going to need if we're really going to solve the problem of cancer. Okay, so let's talk about flies and cancer. Some of you may be surprised to learn that invertebrates, as well as vertebrates, get cancer in the wild. And that's true of Drosophila as well. If you look at aged fruit flies, some of them are about 5 or 10% of them will have intestinal tumors. In the lab, we don't wait for these intestinal tumors to just spontaneously occur. Like mouse models, we genetically engineer them using tissue-specific expression of oncogenes. And I'm going to show you here a model that we use in my own lab. This is an ovarian carcinoma. What you're looking at here is a Drosophila wild-type ovary. And each of these little units is called an egg chamber. It's going to give rise to a single egg. And these are just beautiful little regular organs that are simple epithelia surrounded by a basement membrane. And they're very well organized in wild type. But if we use these inducible expression systems to express 2 oncogenes that are oncogenic both in flies and as well as in humans, what we see over time is the progression of these cells to be transformed into these disorganized, perpetually overgrowing cells that break through the basement membrane and crawl onto other tissues. So these malignant cells in the ovarian carcinoma are just one model. I think you'll hear from Norbert about his work in intestinal carcinomas. I think you'll hear from Wu Men that all of these tumors can be allografted easily into other hosts as well for convenience. So there's a number of different Drosophila models that have distinct advantages. Okay, but what are these fly tumors like? Are they really like human tumors? And the answer is yes. Fly tumors show many of these classic hallmarks of cancer that we know about from the famous Hanahan and Weinberg review. So the fly cells, when they're transformed, are essentially immortalized. They overproliferate indefinitely, no matter what their environment is. They lose epithelial and cytological organization. As I mentioned, they invade through basement membranes, and they become significantly undifferentiated or dedifferentiated. They also show these kind of higher order kind of characteristics of tumors, such as cooperation with multiple oncogenes to make the tumors even more malignant. This example in the middle down here is basically the fly version of neovascularization. So fly tumors, like human tumors, grow so they get hypoxic, and they need to recruit their own oxygen supply to continue to grow. Flies don't have a blood vasculature system, but they do have tubes that bring in oxygen from the environment. And the fly tumors recruit these oxygen-carrying tubules to provide oxygen to them through signaling pathways that are very similar to neurovascularization in mammals. And finally, and critically, these fly tumors, like human tumors, are lethal. So in our models, they reduce the fly lifespan by about 50%. Okay, so these fly tumors really resemble human tumors in a number of ways. But they are by no means identical to human tumors. I want to emphasize that. I've listed some of the differences here, and I think we can talk about some of this in the discussion. But I, again, just want to remind you that there's this incredible track history of Drosophila research and making not only contributions to fundamental biology, but specifically to cancer-related biology. And so I'll close this first section of my talk just with this reminder of this phrase we all know, that all models are wrong, but some are useful. And we think that the fly is a very useful model for studying certain aspects of cancer biology. The aspects that we're most excited about studying, using the fly to study in cancer biology, in my lab, are not really studying these kind of autonomous transforming hallmarks of cancer from the review 20 years ago, but from this new appreciation from the recent reviews that cancer is really a systemic disease that happens not just from the mutant cell, but from interactions between the mutant cell and the wild type cells that surround it. And this kind of view of cancer as a systemic disease that involves tumor-host interactions, we think the fly is really ideally suited for, because it's this simple in vivo system where we know so much about the physiology and the genetics of the animal. And it's simple with all the advantages that I told you about before, incredibly manipulable, and we can use rigorous experiments to ask the general question of how does any animal deal with a growing malignant tumor, regardless of what the particular genetic constituency of that growing tumor is. And we can ask this both from the tumor side, asking how does the tumor negatively impact the host tissues to kill it? And we can also ask how does the host tissue respond to try to fight resistance and tolerance to the tumor? And we think that the fly, because of its advantages, can give us unappreciated insight that we're not getting from these other vertebrate and mammalian models. So what I want to do in the last 5 minutes of my, this history of tumor-host interactions studies in the lab really dates back about 10 years ago when we published a really nice paper back-to-back with Norbert, making the surprising discovery that, again, flies can not only get tumors, but they can show tumor-host interactions that are very, very familiar from human patients. And so one of the best-known tumor-host interactions is cancer cachexia. This is this wasting disease that fat and muscle melt away and is responsible for the death of about 1 in 5 cancer patients directly. I think you'll hear Norbert talk about more this in his own talk. But I'll just emphasize that in our ovarian tumor model, we again see the fat in a control host just wasting away as well as the muscle. And using the rigorous genetics of the fly, we can rapidly identify a molecular mediator. This is a conserved regulator of insulin signaling in the IGF-BP protein family. It's an antagonist of insulin signaling that basically causes insulin resistance. And this molecule has recently been implicated in several cases of pancreatic cancer. In my lab, we haven't stuck with cachexia. We've gone on to look at other host interactions. And a few years ago, we showed that flies also mimic a second major cause of systemic death from cancer, which is clotting defects. So these coagulopathies, which are seen in many human patients where they throw DVTs, are also mimicked in flies, where you see this incredible hypercoagulation, specifically in the tumor-bearing fly, that's then actually followed by a hypocoagulation. And again, we've been able to identify the molecular mediators, which are factors produced by the tumor. Our work has continued to show other cancer-relevant phenomena, but I want to emphasize that this isn't work just from my lab. There's work from many labs. And I want to take the opportunity to highlight 2 recent high-profile papers, one from Norbert's lab and one from Wei Song's lab in China, where they started with a mouse model and then identified conserved molecules that are implicated in both preclinical mouse models of cancer, as well as in some cases with human patients. So the fly is really a great model to study these conserved recognized human tumor host interactions. Before I leave the second part of my talk, I want to just say how we flipped the script a little in my lab, and also asked whether the fly can be used to discover unappreciated and new tumor host interactions in the fly. And using the fly as a discovery system, we found that the inflammation from the fly tumor actually causes an opening of the blood-brain barrier. The fly has a blood-brain barrier that usually, like the human, protects it from circulation, but in the presence of the tumor, circulatory elements drain into that brain parenchyma. And this is directly involved in killing the fly. We collaborated with mouse modelers to show that at least in one mouse model of cancer, the same is true. This is IL-6 mediated, and again, it contributes to the demise of the mouse from the tumor. So the fly can be not only used to explore known human tumor host interactions, but also to discover new ones. Okay, so in the last third of my talk, I want to talk about one of the most exciting tumor host interactions in all of cancer biology that I haven't talked about yet. And that is, of course, the interactions between tumors and the host immune system. So flies have a very robust immune system. They don't have adaptive immunity, so they don't have T and B cells, but they do have an innate immune system. That was actually the subject of the 2011 Nobel Prize. And what I'm here to tell you is that flies not only can make tumors, but they also have an immune response to cancer that fights the tumors. This is not a discovery that my lab has made. This was discovered almost 20 years ago in a beautiful paper by Pepe Pestre-Perea. I'm just showing you data here from our ovarian tumor system, where you can see in a control ovary, a wild-type ovary, the cellular immune system doesn't really, these macrophages don't really recognize the tumor, or sorry, the host organ. But in the presence of a tumor, these macrophages just flock to this tumor, and there they have anti-tumor activity. And we know that because if we eliminate those macrophages from the tumor-bearing fly, we can use this tumor grading system, kind of similar to a pathologist, to see an increase in these high-grade red tumors, again, in the absence of these macrophages that fight the tumor. We can do the same with the humoral immune system. We can compromise humoral immunity. And again, we see an increase in the severity of the grade of the tumor. So the host immune system of the fly is really involved in fighting the tumor activity, analogous to what we see in the human context with anti-tumor immunity. Anti-tumor immunity is conserved in invertebrates. Okay, so we've been really excited about this. We've been studying this in several ways in our lab. And the last thing I just want to tell you about in my time today is how we've used one of the classical approaches in Drosophila, one of the classical strengths, a genetic screen, to identify factors that regulate this. And so we've looked for factors that are upregulated in the tumor tissue and are secreted and produced to the host environment. And these could be things that could regulate interactions between the tumor and the immune system. And then by easily knocking down those factors in the tumor, we can identify those that have an effect on tumor progression or lifespan. And we've identified, in addition to some expected factors, two very poorly characterized factors called TEP3 and NIMB4, that when we knock them down, they extend the lifespan of the tumor-bearing host, and they do this because they slow tumor progression. We get many more of these low-grade tumors here. And I'm telling you about these molecules in this last part because it turns out that both of these regulate the interaction with the host immune system. TEP3 is a conserved protease inhibitor that when we knock it down, it causes a massive increase in the number of macrophages, the cellular immune system that associates with the tumor. And so TEP3 is normally allowing the tumor to hide from these macrophages and prevent their anti-tumor activity. The second molecule, NIMB4, also allows the host to hide from the immune system, but it doesn't do that by affecting the number of cellular macrophages on the tumor. It instead does this by affecting the humoral immune system. And so it's preventing the humoral immune system from killing those tumor cells. NIMB4 has a conserved phosphatidylserine binding motif, and phosphatidylserine is a lipid that is usually localized to the inner side of eukaryotic cells, but is exposed most familiarly in dying cells, but also in stressed cells that include the human tumors and their immediate microenvironment. And so what we think is that this phosphatidylserine binding protein produced by the tumor masks that phosphatidylserine binding, essentially letting the tumor evade the humoral immune system and preventing its tumoricidal activity. So in sum, what I've told you is that we've used these genetic screens and the genetic power of the fly to show that flies not only show anti-tumor immunity, but the tumors can also display immune evasion, and that they can create a cold tumor microenvironment, which is one of the major obstacles for current cancer therapy. And so we're really excited to extend these studies and see how they could inspire us to kind of like warm up the tumor microenvironment, at least for the innate immune system, and potentially help patient treatments. And finally, I'll just end by really emphasizing this remarkable discovery that I think we've made as a field, that not only does cancer, the origin of cancer, predate the split of the invertebrate and vertebrate species 500 million years ago, but these anti-tumor defenses that animals have evolved have also predated that split. And so we think that there's a tremendous amount to learn from this underutilized system in cancer biology. And so I'll end just by thanking the people in my lab that did the work, the funding sources, and I'll turn it over to my colleagues for the next talk.
Okay, so I guess, let's see. Show the screen. Can you see this? Yeah? Can you
Just
hear me?
gotta put it into presenter mode, and then we should be good. Looks
There you go.
great.
Okay, great. All right, so I'm going to be following up on some of the same theme as what David was talking about. But first, yeah, I'd like to thank Justin for putting together this pretty interesting webinar. And so what I want to do today is really to focus on the different methods that we're using to really take apart those Drosophila cancer models. And so what we do is that we're basically using genetic screening, as David was mentioning, but we're also combining this with both transcriptomic, and I'm going to be talking about single nuclear RNA-seq, as well as proteomics, like proximity labeling and metabolomics. And those methods, together with sophisticated genetic approaches, which allows us to generate animals with very complex genotypes, really are helping us to get really novel insights in the processes of tumor formation and interorgan communication. So the system I'm going to be focusing on is the one that David mentioned briefly, where we're generating, in this case, tumors in the midgut. And the way we do this is that we're driving an oncogene. This is a fly oncogene called Yorkie, which is the orthologs of YAP. And we're expressing this activated form of this oncogene in the stem cells in the gut. And where you can see that within like 4 days, so the tumorous cells are labeled with GFP here, and within 4 days, you can get a very large tumor in the gut. And those tumors are quite reminiscent to the human tumors, or mammalian tumors. Is that at least, for example, if we simply look at the metabolism, those tumors do consume a lot of glucose, which through aerobic glycolysis is transformed into lactate. So it's also known this Warburg effect. So we like this model here of the fly gut tumors because in addition of the tumor itself, there are a lot of systemic phenotypes which are being associated with the tumors. So in addition of the gut tumor, the organs are going to be, most of the organs are going to be wasting in the animal. So the muscles are going to be falling apart, the fat is going to fall apart, and the ovaries in females and so on. And so so we think that this is actually a nice model for studying organ wasting. Also that we like to think that this is a model relevant to cachexia that David mentioned. So in addition of those organ wasting, there is a lot of metabolic abnormalities, like there is a depletion of the triglycerides and glycogen, and those flies are hyperglycemic. So we've been performing a number of different genetic screens to identify factors, and here I just want to emphasize that we do have in the fly community RNAi reagents or guide RNAs for CRISPR, which really allows us to do either loss of functions or gain of function studies. So we can simply, for example, interrogate all the, let's say, all the secreted proteins which are produced from one organ and they would affect other organs and so on. So by performing a number of genetic screens in the context of this those gut tumors, we're able to identify 3 different factors that are derived from the tumor that affect the other organs. So one of them is this AMPL2, or an IGF binding protein that downregulate insulin signaling. That's the same that David was mentioning. Then we have a UPD3, which in flies, which is the orthologs of IL-6, which regulate the JAK-STAT pathway, which is also produced by the tumors. And then we have a PDGF-VEGF known as PVF1, which also is derived from the tumor that regulate MAP kinase or TUN kinase signaling in the peripheral tissues. So over the past few years, we've been really trying to understand how those different factors affect the different organs. And I'm not going to have time to go through this in details, but we're starting to build a map of all those different factors work either together or alone in terms of regulating different aspects of the metabolism or the maintenance of the other structure. So so in addition of the classic genetic screen that we can perform to identify factors, in the last few years, we've been really trying to evaluate how we can use single cell RNA-seq. In the fly, we do single nuclear RNA-seq to really study more globally the interaction between the different tissues or tumors and peripheral organs. And so we do have, as a result of this community consortium effort called the Fly Cell Atlas, we have actually the annotation of all the different cell types in the adult fly. And so now we can actually take this information and study, try to understand the described phenotypes at the global level using single nuclear RNA-seq. So this is what we've been doing in the lab in the context of those gut tumors. So you can see here with the GFP at different time point, you see the growth of the tumor, and then the flies eventually, as time goes, develop those cachectic phenotype, and which also involve this very large bloating of the abdomen due to water retention. So we performed a single nuclei full body RNA-seq at different time points. And this is actually just to illustrate here how comprehensive the maps that we're getting are. And the point I want to make here is that it's actually the very doable experiments, because here we simply only like 10,000 or so nuclei, we can really get a single cell, single nuclei RNA-seq body map of the animal. So now we can really use this approach to then identify misexpressions or the perturbed ligands and so on. So just to give you an example that we recently published, we found that in those flies which have those gut tumors, we saw an increase in the expression of 2 different enzymes, PepsiK and PDK, in the flies which carry those gut tumors. And those are overexpressed in the fat body. So they're usually expressed at some level in the fat body, but we can see a large overexpression and a large increase in the flies with tumors. So those 2 different enzymes, PDKs and PepsiK, drive gluconeogenesis. So this is where you go basically from pyruvate acetyl-CoA into the trehalose or glucose. So in flies, most of the circulating sugars are trehalose. And you can see that in the flies here, there is this, at day 8, there is a huge increase in the trehalose level. So we were able to show that that increase in gluconeogenesis is actually important for the cachectic phenotype of the flies. And here what we do, this is an example here of how we can generate animals which are very complex genotypes. What we do is that we generate flies. So we use 2 different binary systems, the Gal4-UAS system for one tissue, and then the Lexeop system for the other tissue. And so here we've generated flies which have gut tumors, but we've removed, in this case, the PepsiK from the fat tissues. And so what you see is that those flies, this bloated phenotype, which is present in the cachectic flies, is being suppressed. And then we see a decrease in the level of circulating trehalose. So then, so the idea then is that there is a factor coming from the gut tumors that regulate gluconeogenesis in the fat tissues. And so what we've done is to use, we've developed over the years an algorithm called FlyPhone, which is very similar to some of the algorithm like cell chat or cell phone people have used in mammals. But basically, it allows us to try to connect the expression of ligands and receptors and pathways across tissues. So using this FlyPhone analysis, we were able to make predictions about specific pathways which could misregulate the expression of those gluconeogenesis enzyme. And one of the candidate pathway was the JAK-STAT pathway. And so it predicted that, again, one of the predictions is that one of the ligand, UPD3 from the gut, would regulate JAK-STAT signaling and affect gluconeogenesis in the fat tissues. And then we're able to test this experimentally by generating, again, animals where which have those gut tumors, and now we remove JAK-STAT signaling by RNAi in the fat tissues. And we're able to demonstrate, in this case, again, that the level of trehalose and the gluconeogenesis pathway was downregulated when we remove JAK-STAT signaling from the flies with gut tumors. So this is a model that we developed then, that the fly tumors produce this IL-6 UPD3 activate JAK-STAT signaling that leads to an increase in PepsiK and PDK, and that leads to an increase in gluconeogenesis. So we were actually fortunate to be part of a consortium called the CanCan from the Cancer Grand Challenge. And that involved a number of different groups, some of them that study mouse, that study cachexia in mouse, and some groups that study cachexia in humans. So we were able to take the observation that we made in flies and now ask whether or not this regulation of gluconeogenesis was also affected in different cachexia mouse model. So in this case here, in one of the cachexia model, we're able to demonstrate that there is an increase in PepsiK and PDK expression in the cachectic mice. And in this experiment here, a set of the cancer cells were injected into mouse, and those cancer cells express IL-6, and so there were two different experimental setup. One which is simply cancer cell expressing IL-6, and then another set where there was an inhibitor called PS10 of PDK, and the mice which had the transplanted cells where PDK was inhibited, actually in this case survived longer, suggesting that the role for this gluconeogenesis pathway is also important in those mouse models. In addition, also we looked in patients, in humans, for a correlation between the level of expression of PepsiK or PDKs in human, and there were an increase, again, a nice correlation between the level of expression of those different enzymes and the severity of the cachexia phenotype. So the take-home message then is that we think that both in fly and in mammals there is this IL-6 JAK-STAT connection is present, and that may be involved in the regulation of gluconeogenesis. So I have told you about how we've been using here single nuclear RNA-seq to really mine the data set, so we understand, come up with hypotheses about specific misregulation that could explain some of the organ wasting phenotype. Now I want to illustrate that we're using proteomics, and in particular proximity labeling, as a way to identify additional factors involved in inter-organ communication. And here the method we use here is proximity labeling using a promiscuous enzyme called the biotin ligase called BIR-A or turbo-ID. And the idea here is that we express a promiscuous biotin ligase in the secretory pathway of one tissue. So in this case, for example, just in the fat. And then the idea is that those all the secreted factors are going to be labeled with biotin, and now we can dissect or separate the other tissues that we are interested in. Let's say, for example, the muscle, and then isolate using streptavidin, all the biotinated protein, and then perform mass spectrometry. So we've been using this method actually quite extensively for, like in this case, to identify all the tissue of origin of all the proteins in the fly blood. And so in this experiment here, we used a number of different GAL4. This was done in larvae, where we express the BIR-A either in muscles or inocytes, which are the hepatocytes or the fat tissues and so on, and then we bled the larvae and identified many of the secreted proteins which are present in the blood. And this really gave us a resource of all the origin of the different proteins in the fly blood. So we identified about like 540 proteins, and some of them are produced only from one different tissue and other organs, or some from multiple. But this really gives us now a list of parts that then we can go back and interrogate in the context of the different models of whether or not some of those would be overexpressed or decrease in different cancer models. I should say that in addition, we've also more recently been using this proximity labeling approach in the context of those Yorkie and gut tumors to identify factors which are secreted by those tumors. So I'm going to stop here, but basically what I wanted to do is to illustrate how we are integrating a number of different approaches from transcriptomics, proteomics, metabolomics, as well as using the genetic tools that we have, like based on RNAi and CRISPR, to really identify factors which are involved in the communications between tumors, in this case, and the other peripheral organs. And then I also wanted to illustrate how we're using different binary systems to generate animals which have a complex genotypes, which to really allows us to really test the role of the different ligand receptor pairs in the context of, in this case, interorgan communication. And yeah, I just wanted going to leave that here, but those are some of the main people that were responsible for the work that I've described, as well as some of our collaborators on the proteomics and the mammalian experiments. So I will pass it on to Wuming. Let's see. Stop share.
Okay, I can share now. So let me just do the presentation. Okay, so can you see my slides?
Yep, looks great.
Okay, thank you. So I also like to thank Justin for organizing this webinars, and thank David and Norbert for the wonderful introduction of the fly system and the techniques used for cancer studies. So I'm going to tell you some of the studies in my lab that we established several tumor models to address challenging questions in cancer biology. So I'll start with this slide that many of you probably remember this provocative questions initiative launched by the NCI probably about 15 years ago. So many of those questions I checked actually trying to understand how cancer get initiated. And around that time, so there was a talented postdoc, Yoichi Tamori, who is a faculty member at the University of Louisiana right now. So he was interested in working understand how the tumor suppressor gene knockdown or mutations would actually lead to tumor formation. When he looked at those neoplastic tumor suppressors, he found that not all mutant cells created equal. So basically, when you create those knockdown cells in the wing pouch region, so this is the wing imaginary disc, so those mutant cells actually gets eliminated through a biomechanism called cell competition. But if those knockdown or mutant cells are generated in the hinge region, so those actually become tumors. So the reason behind that is actually there is endogenous JAK-STAT activation in those regions we dubbed as the tumor hotspot. So this actually led us to propose this concept called tissue tumor hotspots. Basically, to get the tumors to get started, you not only have to have the seed, which is the cells bearing those cancer-related mutations, but you also need to be in the right environment. So basically, you have this seed and soil concept to get the tumors to be started. From then, we started to looking at to see if there are other regions, other tissues have also had those tumor hotspots. When a talented former graduate student, Shen and Yang, joined the lab, so he was actually able to identify a very interesting group of cells. So this is actually a lower salivary gland. So so there is a very small group of cells that are located in the imaginary ring of this at the boundary between those precursor cells. These are the lower imaginary ring cells, which are the precursor of the salivary gland, but also between these giant salivary gland cells. So these cells actually are specifically prone to tumorigenesis. When we give them the oncogenic notch activation, then the tumor is only formed in these group of cells. So we created another interesting model, which, this we call this transition zone tumor model, which has precise location. So we know exactly where those tumors get started. And also it has defined number of tumor initiating cells. So only those transition zone cells will become tumors. And also it's highly reproducible in tumor induction. So it's actually not surprisingly, so the region that allow these cells to become tumors also enriched in the JAK-STAT signaling, just as I showed you in the wind disk model, and also the JNK pathway is also activated. So, so this transition zone concept is actually not an invention in the flies, actually in the mammalian system, human cancer biology. So people have identified those esophagogastric junctions, also the endo-ecto-cervix junctions. Those are where the two types of epithelium meet. So they are the regions that actually highly prone to a type of pre-malignancy called metaplasia, and which one of the famous examples is Barrett's esophagus. So we basically created this transition zone tumor model to understand how tumors get started. So when a postdoc, Xianfeng, joined the lab, so he was able to show that actually these transition zone cells actually are polyploid cells. So the reason they become tumors is that when notch gets activated, force these polyploid cells to re-enter mitosis, and then they become malignant, transformed into tumors. And we can see those polyploid cells actually undergo a variety of different types of cell division or the cell cycle programs, including the interreplication to become even larger cells, or some cells, those polyploid cells, will undergo EROM-prone mitosis, and also some will actually go through a process similar to the meiosis, depolyploidization, basically produce lower ploidy cells. So basically, this tumor is actually a heterogeneous group of cells with various different types of cells, with tumor cells with different ploidy levels. So this actually resembles the human cancer group studies actually revealed that it's a polyploid giant cancer cells, PGCCs, which originally identified by Dr. Jason Liu from MD Anderson. So, so this actually, some of the these polyploid cancer cells in flies actually resembles how the these polyploid PGCCs actually gets divided, and they're very similar. So this, we can also use this model to understand how the PGCCs actually promote tumor progression and get started as well. So when Xianfeng continued to using this model, he noticed that the tumor size actually are different between the male and the female larvae. So we know that the the salivary gland is not a sexual organ, and then the transition zone cells between male and female in the control situation, they don't differ that much. They have about the same number of cells, and the cell size is not different. But then when tumors get started, then the females actually grow the larger tumor, which is about 2.5 times higher than the larger than the male tumor. So this raises a very interesting question: how the tumors show this sex dimorphic growth. So actually, turns out Xianfeng was able to identify the JNK signaling actually is different between those two sexes. So the females actually had slightly higher levels of JNK activation based on the reported analysis. We can block this pathway, using the RNAi lines against the canonical pathway components, and then you can see the tumor size, the sex differences eliminated. So we're curious, what is the origin of the how the JNK pathway gets activated, so we perform a single-cell RNA-seq analysis, we were able to identify a group of cells. Actually, the one that David just mentioned is the hemocytes or the macrophages. So they're actually become part of this tumor microenvironment. So this is also a good model to study how tumor microenvironment influence the tumor growth. So those hemocytes, macrophages, actually carries, so they're enriched in this IGA, which is the ligand of the JNK signaling. So it's a TNF alpha homolog. So the females actually has slightly higher levels of the IGA signal. So you can see that they're expressed in the hemocytes. And then we can reduce half of the IGA in the fly, and then the tumor size and then the sex dimorphism is also reduced. So then we can also use this very specific LEX-A system to knock down the IGA in the hemocytes between male and females. You can see that now the sex difference is also reduced. Then as a control, we can see that the IGA from the fat body, the other potential source of IGA is does not affect the tumor size. So most interestingly, Xianfeng was able to manipulate the sex determination pathway of these flies. So basically, we can change the hemocytes from the male to female or from female to male while you're producing the tumors. Now you can see that when we produce those male hemocytes in female larvae, now the tumor size is smaller and also the sex difference is gone. Then when you have the female version of the hemocytes, then they actually promote the tumor growth even larger than the male tumor, actually larger than the female tumors. So from this study, we can see a very delicate regulation. It's complex, but also involves multiple organ into organ communications. But at the root of this sex dimorphic growth is those hemocytes, which is actually immune cells in the flies. So Xianfeng was actually able to identify a more complex, because the JNK activation in the tumor actually leads to the UMPEL-2, which is the functional ortholog of leptin. So the UMPEL-2 in the tumor actually cause the release of the Drosophila insulin-like peptides from the brain, which the insulin-producing cells, and that actually activates different levels of the insulin signaling in the tumor. So you have this, recruiting the hemocytes, then involving the insulin-producing cells to regulate the overall size of the tumor growth. So another advantage of this tumor model, David mentioned a little earlier, is that to allow the allograft, so the transplantation of those tumors. So we can surgically remove those tumors from their original location and then transplant them into the abdomen of the adult fly abdomen region. So you can see the tumors will grow there. And then this process, actually, you can repeat. So basically, you can take those tumors out and then cut them into smaller pieces and then have this repeated transplantation. Then the longest we have for those tumors is more than 2 years now, after 70 rounds of tumor transplantation. So then we can use this to do a lot of analysis, including the tumors and also the host flies, to understand the tumor-host interactions as well. So it's a very interesting thing we found is that while we're doing this repeated transplantation of those tumors, the high passage tumors becomes more malignant. So basically, they could cause significant damage to the host. You can see the host lifespan becomes shorter comparing to those hosts with the earlier generation of those transplanted tumors. Of course, we also observed different phenotypes as reported from David's and Norbert's talk, that you can see there's bloating, also the organ wasting in this tumor model. So so when Fei and Hong Cun decided to look at how this tumor will affect the organs, they focused a very interesting tissue, the organ is actually the fly kidney. So these are the nephrocytes. They're the functional homolog of the podocytes. They can filter the human lymph of the fly host. You can see the tumor host, actually you have a lot of damage of the these nephrocytes. This is a slit diaphragm. You can see the control has a much organized finger print-like pattern, but then you have a lot of aggregates in the tumor hosts. And then one of the things that we looked into is what really caused this tumor host response. So Fei was able to identify is the immune activation. So as David and Norbert mentioned earlier, so you have fly has this innate immune response. So the TOL pathway and also another pathway called the IMD pathway. So in this case, the IMD pathway is activated in the nephrocytes. You can see the reddish become nuclearly localized. And then we can mimic this situation by activating the IMD pathway to also cause the nephrocytes damage. And then if we knock down this IMD pathway receptor, PGRP-LC, then you can see the nephrocytes morphology is improved, and also the lifespan is also improved as well. So then Fei was curious to see how this IMD gets activated in the tumor host. It turns out that these flies, the tumor host actually has dysbiosis. So the gut microbiome gets, there is a particular type of gut microbiome called Acidobacter is significantly upregulated. So this is a gram-negative bacterium, and then this is a potential activator of the IMD pathway. And then Fei was able to take the human lymph out from this host and then plate on the plate. You can see that then there's actually bacteria in the fly's host's human lymph. And then if we reduce the bacterial load by raising those flies with antibiotic food, now you can see the nephrocytes morphology is improved, and also the lifespan is also improved. So we can also do a mono association when we have those germ-free fly hosts, and then if you raise them with either Acidobacter or the Lactobacillus, you can see that Acidobacter causes very significant damage to the nephrocytes. And so more significantly, if we feed these flies with a kidney drug called AST-120, which is a human kidney drug, and then you can also improve the morphology of the fly nephrocytes and also increase the lifespan of the tumor host. So this book actually just go online today. So if you're interested, it's an EMBO journal, so you can check this out. So because of this very significant immune response in the tumor host, we also started a collaboration with Dr. Hongjae Lee's lab at Baylor University. So we actually identify the tumor host tissues, and also obviously the tumors are there, and then we can see actually both tumors will affect the host cells, host cells would also affect tumor cells as well. So I hope to be able to report that to you at some other point. So to summarize, I've showed you that the insights from our tumor model studies, exist as tumor hotspots, and then some of those tumor hotspot cells are polyploid cells, and then they can contribute to the tumor initiation and progression, and also there's a tumor microenvironment, a systemic regulation of tumor growth, and also notice that bacterial translocation and the immune response has been related to the tumor host response as well. Lastly, I would like to tell you that we have been there's a book called Drosophila Model in Cancer. That the first volume was published 2019. So there's a new one coming out, volume 2, with myself and Tana as the co-editors. So you can check it out as well. With that, I'd like to thank the funding sources and then people who did the work and our lovely FlyBio community. So yeah, so I think I'll pass the to Justin. Let me see, I can stop this.
Thank you, Dr. Deng. Should be like an orange button that says stop sharing on top of the screen.
Got it. Okay. Thank you.
All right. Well, first, I want to thank all of our speakers for those wonderful talks. I think we saw a really great swath of Drosophila cancer models here, and I'd love to discuss a bit more. I have a few questions that I've been asking at all of these webinars just to keep things standard. And the first question that I hope everybody can answer just briefly, because we are on a little bit of a time crunch here. What is unique about Drosophila that makes it useful as a cancer model? And I know that you already talked about a bunch of these things in your talks, but maybe just something that you didn't touch on or something that you think is especially important. Go ahead, take it away, Dr. Bilder, if you're okay going first.
Sure. I mean, I think Norbert gave a great example of the complex genotypes and the pioneering biochemical and other techniques that can be used. I think the thing that I would add to that is just that, flies really are rapid, cheap, and easily manipulable. And so I think they're kind of a little bit, you could think of them as we can get them in big numbers where we're doing experiments on hundreds of flies rather than, 4 or 5 vertebrate animals. And so I think from the NCI's perspective, I think you could think of them as kind of a not low risk, but a low stakes model to invest in for NCI that can be a real hypothesis generator for animals that can then lead to mammalian validation. So I think that that's a really unique aspect compared to the mouse.
Yeah, I would just add that, I mean, the scale at which we can do experiments is really, quite unique. Like, again, we can simply identify, let's say, take, 500 or so putative secreted protein from one tissue, and within a couple of months, a few months, you can test them all in the paradigm that you're studying. So I think that's, and again, also the single nuclear RNA-seq, the full body thing. I mean, it's not like you can take a mouse and just do the full body, map of all the different cell types. But in flies, you can do phenotypic analysis now at that scale very quickly.
Yeah, I think that's summarized really well. Yeah, you can not all the organism you can do this, single nuclear whole body sequencing to get all the organs response at the same time, and of course then the statistical power you can actually generate. For instance, in our case, we can generate this tumor model is very reproducible. You can get hundreds of tumors in a very short period of time. And of course, all those powerful tools, you can do the genetic manipulation. You can actually separate the host and then the tumor, right? You can manipulate the host, allograft model. You can do the genetic manipulation separately between those two parts.
Great, thank you. Second question is, what are the most important differences to keep in mind between Drosophila cancer and human cancer? David, I know you brought a couple of these things up in your talk. Feel free to jump in, whoever wants to go first.
I can just jump in again to maybe just to get things started. I mean, I think it's really important to keep in mind that these are different, creatures. And so there's a lot of similarities in the anatomy, but there are important differences, like the difference of the absence of an adaptive immune system, the fact that metastasis is not a major player in the models that we use so far. But I think in all of these cases, what we're seeing is if you don't go on kind of, strict homology in terms of limitations, but think more about conceptual themes about how an animal is dealing with a tumor, you're seeing really important parallels. So if you get beyond kind of a narrow-minded model of limitations in terms of the exact molecules or the exact anatomy, and keep in mind more of these themes, I think there's a tremendous amount that can be learned from that.
Yeah, I think another important difference is really the complexity of the cell types of the tumors. What we're dealing with, like when we are looking at those gut tumors, we basically have like 3 or 4 different cell types. If you're looking at in many of the cancer models in mammalian system, you have many more different type of cell types. So that's something we need to keep in mind here.
Yeah, that's also I felt as well. For instance, like this tumor microenvironment, so we can see the macrophages go there. But then in human tumors, you have a lot of other types of immune cells. So so we're also kind of interested, those macrophages, when they got into the tumor microenvironment in flies, do they also differentiate into different ones? But I guess the complexity is much less comparing to the human situations.
Okay, and the final question before we move on to some audience questions. What are the major obstacles to more cancer researchers adopting this model for use in their labs, in your all opinions?
Again, just to start the conversation, I think there's, there's awareness and this idea that insects don't, get tumors or can't get tumors in informative ways. I think there's a tremendous amount of, not unfamiliarity with what the fly has donated. Every fly researcher has encountered a mammalian researcher who doesn't know where the notch protein came from or where the name hedgehog came from, that these fundamental discoveries were made in flies and are absolutely, 100% relevant to humans. So I think that is that kind of like conscious or unconscious bias is a real obstacle for people reaching out. But I would really point out that, as Norbert emphasized in his talk, there is a tremendous amount of community resources that are freely available and information. And Drosophila researchers are really eager to partner with human clinical researchers or researchers in preclinical models to share their experience and to find synergies where the two systems can inform each other.
Yeah, actually, I think that one of the major obstacles is funding. And I'd be interested in finding out actually from you, Justin, how many fly grants does the NCI support actually?
Yeah, so I think another thing maybe for others to adopt the fly model is the genetic tools may take a little time for the other the researchers to learn. But I mean, fly researchers are very good at sharing. So if you reach out, we definitely will be able to help.
Yeah, and maybe just to re-emphasize what Norbert said about funding. I mean, I think I've looked it up and I feel like maybe there's only one or two in the entire NCI budget, maybe only one or two grants that have significant fly components. And so my personal hope is that this webinar series might raise awareness of that and cause NCI leadership to kind of reconsider the value of some of these models.
All right, thank you all. That's really interesting answers to each of those standard questions. And now we can move into some of the audience questions. And unfortunately, I don't think we have enough time to get to all of them. If you have one, I'd recommend just throwing it in the chat, and I'm going to take the questions from the chat and repeat them here. So first question comes from Somia, who asks for Dr. Bilder. Does the humoral immune system in flies work similarly to the cytotoxic immune cells in mammalian tumors? Or in other words, how does tumor killing happen in flies?
Right. So the humoral immune system works similarly in terms that it lyses the tumor cells. It does this primarily, the fly humoral immune system primarily does this by a group of proteins that are called antimicrobial peptides. Antimicrobial peptides are a class of proteins that are also found in humans, where they control the human microbiome. And antimicrobial peptides have been investigated and demonstrated to kill human cancer cells as well. And they do this by forming pores, probably pores that lyse the cells on the negatively charged membranes, which is part of the reason for this phosphatidylserine binding that I talked about. Okay.
This next question is for Dr. Perrimon from Tulane. Sorry if I'm pronouncing anybody's names wrong in advance. Do cachectic Drosophila also have fat and muscle loss as in mammals? If yes, are there muscles that are lost more quickly compared to others? For example, flight muscles or like in humans, leg muscles.
Yeah, I mean, the muscles are falling apart. I mean, the fat also is disintegrate. So it's really a very systemic effect. I mean, we also have, those flies also have a problem with their Malpighian tubule, their kidneys. So it's it's a very global wasting of all the tissues. Whether or not so most of the studies we've done are actually in the indirect flight muscle because they're a lot easier to work with. And we would have to look at the other muscles in more details, like the leg muscles and so on. And it's possible, in fact, that they may not degrade as quickly, the leg muscles, because their metabolism is quite different. So I would expect some differences, but at least our lab hasn't really looked at this carefully.
Okay, question from Jose for Dr. Deng. Do you see an increase in Smurf flies during cachexia? Is the increase of circulating bacteria due to a leaky gut, or is there some other explanation?
Yes, it is. These flies do have a leaky gut. So actually, they not only the bacteria itself, but also, of course, like PGNs, we can also detect higher levels of PGNs associated with there's a peptidoglycan, the associated with the tumors. So yeah, so the answer is yes.
Okay, question for Dr. Bilder from Isabel. Does the TEP3 knockdown in the ovarian cancer model, which causes macrophages to flock the tumor, cause any improved outcomes, like better survival or lower tumor burden?
Yes, absolutely. In fact, that's how we found it in our primary screen, was just by looking for gene products that, when knocked down, would extend the lifespan. And so I think it's about a 30% extension of lifespan when the flies have the TEP3 knockdown in the tumor. And that correlates with the slowing of the tumor progression when we see all these tumor-associated macrophages associating with the tumor.
All right, moving back to Dr. Perrimon. What do you think is so special about UPD3 compared to UPD1 and 2 that allows it to act as a systemic signal? Any evidence that distinguishes the different UPDs?
Yeah, so some of them, like the UPD1, they seem to interact with the extracellular matrix in different ways. So there was a paper from Martin Ziedler a while back, many years ago, showing that they do have different abilities to be secreted long range. That's probably because of the way they are either being packaged or the way they interact with the maybe proteoglycans and so on. But they all do signal for the same receptor, so it's really an issue about diffusion. Right?
Another question for Dr. Deng from Dimitri. Do you detect increase in copy number alterations in tumors that you've transplanted multiple times?
Yes. So when we actually did a single cell copy number variation studies in those transplant tumors, there is a increase of copy number variations and also increased aneuploidies. But the ploidy levels is actually plateaued at a certain point. So the average ploidy of those tumors is not going to be indefinitely increasing. So I think it's a balance of the depolymerization, interreplication, and also the polyper division. So those actually, for some reason, they can stabilize. So the older tumors, actually high passage tumors, may have stronger dedifferentiation. We're still kind of trying to understand if after a long time of passaging, see what happens to those tumors.
Okay. Question for Dr. Perrimon from NAMI. What if I want to identify gene functions that have no homologs in Drosophila related to cancer cachexia?
Well, I guess you can. You can just take some RNAi lines and perturb them from the tumor and see if you see an effect. I mean, we don't work only on the genes which are conserved, right? Or maybe I didn't fully understand the question, but I think this is.
Yeah, and always feel free to add more clarification in the chat if you if we're getting your question wrong. One more question for Norbert. from the single nucleus RNA-seq, sorry, how similar are the tumor cells to normal gut cells?
Oh, there are lots of differences. I mean, I'm not sure we have, I can give you a number, but it's, I mean, there are many differences.
So single nuclear sequencing, those tumor cells are definitely different from the original salivary gland cells. So they are very different, totally transformed.
Okay, and I think, unless I missed a couple in the chat, I think this may be the last one, again for Norbert from Christos. In the dual LEX-A GAL4 system, have you developed a way to generate LEX-A tumors in an inducible manner, like the GAL80TS for GAL4?
Well, the GAL80 works for the LEX-A as well, so you can use it, the GAL80.
Ooh, I think I did miss one at the end here, so sorry, Dr.
So, I
Perrimon.
mean, you can, there are versions of the LEX-A which are repressible with the GAL80, so you can use that in combination to make the LEX-A system TS.
Okay, final question for Dr. Perrimon. Can the biotinylation method be used to identify new proteins in other tissues? And this is from Alexander.
So I think what you're asking is I mean, this is what we do, right? I mean, we label the proteins from the, let's say from one tissue, and then we identify those proteins which have traveled to another one by either dissecting or the other tissue and then doing the streptavidin pull down. That's how we are able to identify circulating proteins.
Okay, and we just got a fresh question from Jose for Dr. Bilder. Do you know what the source of hemocytes getting attached to the tumor is? Are they coming in from circulation, or are they tissue resident? Is there increased proliferation of hemocytes during cancer?
Yeah, that's a great question. That's something we're looking into now. The accepted paradigm is that there isn't any adult hemocyte proliferation, but boy, that would be remarkable if that were actually happening, and that's a possibility that we're actively exploring.
Okay, I'm seeing a few more messages coming in, but we do only have 3 minutes left. So I want to just encourage anybody in the audience to, I'm sure that the speakers would be willing to answer questions via email in the future. Feel free to reach out to them or to me if you have questions. Thank you for all the excellent questions and for the thoughtful responses from all the speakers. Does any of the speakers have any closing remarks that you'd like to, speak out about now before we close?
No, just I think to reiterate that I'm very pleased again that you've initiated this exploration, Justin, of non-mammalian models of cancer, and that I hope it will encourage NCI leadership to consider things like targeted funding opportunities, since it's been a challenge to fund through the standard NCI study sections, these non-mammalian models.
Okay, well, yeah, thanks again for everybody's participation, including the speakers. I hope you learned a little bit about cancer biology research in Drosophila. Be on the lookout for an evaluation survey after this series is over. We'd love to get your feedback on the webinars, and hopefully, possibly we could do some more of these with your feedback. Recordings for all 3 webinars in the series will be available on the DCB events website, so be on the lookout for that as well. That will be in the same email as the evaluation survey. And everybody have a great afternoon. Thank you so much for participating today.
Thank you.
Thank you, everyone.
And Thomas, I think we can stop the recording if you're able to do that.